From cells to fully fledged organisms, from molecules to ecosystems, life exists in a myriad of forms. Until now, biology has sought to observe and understand it, perhaps even to control and exploit it. But today, scientists are looking to invent and manufacture new living beings, basically to synthesize life. This is the challenge of a new techno-science, synthetic biology. Biology 2.0, which promises to solve all our problems. Is this a new and ultimate industrial revolution? Or is it just the opposite? By affecting life in such proportions, are we not taking unprecedented risks? To find out, I traveled the world to meet the field's leading figures, to learn and discover more, and sometimes getting completely lost.
I envision a day when, if you want to produce a particular product, you might custom build a cell for it.
There are so many capabilities. We want to ensure that we leverage only the benefits, and we try to identify any security vulnerabilities or risks to mitigate them.
Since this is going to be so powerful, because this is going to have such wide impacts, let's slow down, let's think carefully, let's put a bit of a pause on commercializing these technologies.
My first surprise is to discover that certain applications are already being studied and are even at an advanced stage. There aren't many of them, but they're very promising. They promise to invent what nature would never have made. Quite something, huh? In any case, the notion immediately aroused my curiosity. I come across one American scientist near San Francisco who is using a yeast, an astonishing unicellular fungus, to produce a molecule to combat malaria. I head for the west coast of the USA. A yeast to make bread or brew beer. And as it happens, my appointment isn't at a laboratory, but in a microbrewery.
Hello. Hi. Welcome. My name is Jay Kiesling. I'm a professor at the University of California, Berkeley. I'm also CEO of the Joint Bioenergy Institute in Emeryville, California. My definition of synthetic biology is the engineering of biology, manipulation of biological systems to accomplish some goal.
Jay Kiesling is one of the stars of this new discipline. As he describes the beer manufacturing process step by step, Jay tells me that his reputation came from his work on artemisinin, a molecule discovered in the 1970s to combat malaria. Until now, this molecule was extracted from a small, rare shrub, which only grows in a few parts of the world. Jay Kiesling has found another way to produce it, by modifying the genome of a yeast.
We've taken the genes out of the plant that naturally produces artemisinin and put them into yeast. And so it's just like brewing beer. You put the yeast in a vat with water and sugar and some other nutrients, and the yeast grows up, and rather than spitting out ethanol, as it would making beer and wine, it spits out artemisinic acid, a precursor to artemisinin. And then we use a process, and it transforms from artemisinic acid into artemisinin.
Fortunately, what Jay gives me to drink is only beer. It all seems so simple. Making a new yeast and investing it with a role that nature had never thought of. It makes my head spin. Or maybe it's the beer. I don't think it was drinking beer that gave Jay the idea in the first place. A short drive away is the University of California campus at Berkeley, where he started out at a time when synthetic biology didn't yet exist.
I was fascinated with genetics. This is why I got into biology and why I took an engineering route. And so when I started as an assistant professor, I realized that we didn't have tools available to readily engineer metabolism inside cells. And so I started projects with my students on developing tools for metabolic engineering. But you can't just develop tools and not use them for something and not prove that they're going to be useful. And I decided, you know, I should better think about some applications for these tools. So we started looking for applications, and we found artemisinin. I knew about malaria from a microbiology course in college. Knew nothing about artemisinin, and we looked into it and we said, boy, I think we can produce this, or at least a precursor to this. At the end of 2004, we had a $42 million grant from the Bill and Melinda Gates Foundation to finish off the process, to engineer a microbe to produce the product and then license it out to a company. Everything moved, in retrospect, pretty quickly. When you're in the process, you think, can't we move this faster? Can't we move this faster? There's people dying. Now, there's a large-scale process for producing it. They've produced on the order of 60 tons of the material, which is enough for 120 million people.
The wager paid off. Engineering life as one would engineer a machine. To understand how this is possible, we have to look at the basic tools of synthetic biology, using and combining the functions coded within DNA. Jay Kiesling first of all reminds me that each organism, each living cell, has one or more functions. Certain cells aid in digestion. Others convert light into energy for plants, or transform sugar into alcohol, like yeast, a surprising little unicellular fungus. But how do these cells know the nature of the work they have to accomplish? They quite simply have a manual or a program inscribed in their nucleus, their genome. The genome is an organism's instruction book for life. It describes all of a life form's functions. The book is made up of several chapters, which we call chromosomes. Chromosomes themselves are composed of genes. They form the sentences in the book. Each gene or each sentence is read like an instruction. All the genes together tell the cell what it has to do. All of this is written using an alphabet, DNA, made up of the well-known molecules A, G, T, and C, for adenine, guanine, thymine, and cytosine, which are always organized in pairs, the A with the T and the C with the G, known as base pairs. Genes are transmitted from one generation to the next identically, or are modified to respond to essential new functions or new requirements for the cell to survive. This is what we call evolution. If we know how to modify them and put them together, we can then change very simple functions into more complex ones. We can then imagine useful combinations that nature never attempted through evolution. With artemisinin, Jay has demonstrated that it was feasible and workable on an industrial scale. He transformed a cell into a chemical factory that produces active molecules. This concept enabled synthetic biology to avoid the problems that previously limited production capacities.
There are many advantages of producing a drug through microbial fermentations rather than having to extract it from a plant. One of the challenges with the plant is that it takes a significant amount of time between the time that farmers get seeds and companies decide how much they really want and contract with those farmers. The farmers get the seeds, plant them, harvest it, get the molecule or the extraction to the pharmaceutical company, and then they derivatize it and put it into the drugs. That process takes somewhere around 14 months to 2 years. And because of those long time scales, there's been large swings in the price and availability of artemisinin. It swung anywhere from about $200 a kilogram to over $1,100 a kilogram. And what that means is that when the price is high, people in the developing world can't afford it. When the price is low, farmers don't want to plant it because they can't make a profit, so they switch to other crops.
Be it Jay Kiesling or myself, or a developing world farmer, we're all the same. Whatever the invention, however avant-garde it may be, we also want it to be profitable. Jay's not only a scientist, he's also a businessman. He set up a company and runs another, JBay, a laboratory co-funded by various companies and the American government. Along the way, has he forgotten his noble ideas about saving the world?
I believe strongly in a well-regulated capitalistic system, but we have this system that gets products out to people. The company won't make any profit off this anti-malarial drug. In fact, and the University of California, which holds the patents to my work, gave the process away for free. We'll make a profit on something else that doesn't have to do with the developing world. I don't think that the non-profit model works for everything, right?
That's right. So we have 7 partners.
What the company gets out of it is a well-honed organism. It produces artemisinic acid, but you can make a few changes in its genetic makeup now and it'll make many other things. And this is really a core demonstration of the power of synthetic biology. You build a platform and it can do many things. It's just like a computer. You can use a computer to write a document, a book, to give a presentation to an audience. You can use it to control the heating system in a building so that it's optimized. You can also use it to control some of the circuits in a car. Computers have many uses, just like some of these platform organisms will have many uses.
Jay Kiesling intends to make money from other applications derived from the same platform that's used to make artemisinin.
It turns out that I can apply that to many things. To producing drugs, to producing flavors and fragrances for ice cream, and for perfumes. I can apply it to producing fuels that would be carbon neutral, to producing the precursors to carpet that is produced in an environmentally friendly way that maybe has different characteristics. So my science is extremely narrow, but I can apply it to many different things.
Listening to Jay Kiesling, I wonder what the future of synthetic biology holds. Will it be the eagerly anticipated new industrial revolution? Some say there's no doubt.
We've got a long way to go. I would say we're still in the very early stages of what we'll eventually be able to do with synthetic biology. Right now, we use microbes that have been demonstrated by industry to be scalable. Right? We can build chromosomes, and people can claim that it's a new life form, but for the most part, they're copying what nature gave us and making some changes in the DNA. Wholesale re-engineering of an organism, that's still many years off. I envision a day when, if you want to produce a particular product, you might custom build a cell for it. I'm hoping that we'll have plants in the environment that can help clean up the environment. Maybe that will sequester carbon from the atmosphere better, or that will degrade contaminants that have been accidentally spilled in the environment.